Enzymes are crucial in biochemical reactions and metabolic pathways that take place inside cells. Enzymes are biological catalysts. A catalyst is something that speeds up chemical reactions. Most enzymes are proteins. The role of enzymes is to lower the activation energy of a reaction. Remember, enzymes bring the activation or the energy needed for a reaction to occur to a lower level so that these reactions can be made possible. In a chemical pathway that has an end product, each of these steps in the pathway is catalyzed by a specific enzyme. If this is a chemical pathway, the starting compound is changed to an intermediate A by enzyme A. Intermediate A will then be changed to intermediate B by enzyme B. Intermediate B is changed to the end product by the last enzyme in the pathway. Controlling any of these enzymes will allow the control of the end product. A specific enzyme facilitates each step of a biochemical pathway. If we want to control the growth of microorganisms, we could inhibit an enzyme in a pathway. Inhibiting the enzyme in a given pathway will eliminate the intermediates and the end products of that pathway. In order to control the growth of microorganisms, we could inhibit enzymes in a metabolic pathway, thus inhibiting the formation of a crucial end product for the cell, which will eventually kill a population of cells. Enzyme inhibitors are substances that block an enzyme's active site without denaturing the enzyme. In other words, enzyme inhibitors will affect the active site of the enzyme. If the active site of the enzyme is affected, the enzyme cannot work. There are two types of inhibitors: competitive inhibitors and non-competitive inhibitors. A competitive inhibitor is a chemical that competes with the substrate for the active site of the enzyme. Remember, the substrate is the reactant that fits in the active site of the enzyme and will be converted into the product for that reaction. A chemical that fits in the active site of the enzyme but is not a substrate is called a competitive inhibitor. Those kinds of substances compete for the active site of the enzyme against the substrate. Non-competitive inhibitors, on the other hand, also called allosteric inhibitors, are going to fit on the enzyme at a site different from the active site. For example, here is an enzyme in green. The substrate is depicted in pink and purple. The enzyme will catalyze the reaction that separates the two parts of the substrate, creating the product for that particular reaction. A non-competitive inhibitor will bind the enzyme at a site different from the active site. The binding of the inhibitor to this site, called the allosteric site, changes the conformation of the active site, and now the substrate cannot fit in the active site. Thus, the enzyme is inhibited or stopped from catalyzing the reaction. An example of a non-competitive inhibitor is fluoride. Fluoride indiscriminately binds to enzymes, inactivating them by changing the conformation of their active site. In doing so, fluoride kills bacteria. A competitive inhibitor is going to be a chemical that has a conformation that is similar to that of the substrate. Both the substrate and the competitive inhibitor could fit in the active site of the enzyme. The competitive inhibitor competes with the substrate for the active site of the enzyme. If the competitive inhibitor fits in the active site, the enzyme cannot work. When the substrate sits in the active site, now the enzyme can change the substrate into the product for that particular reaction. Because the competitive inhibitor competes for the substrate for the active site of the enzyme, if the concentration of the substrate is increased, the competitive inhibitor has less chances to reach the active site of the enzyme. An example of a competitive inhibitor that is used to control bacterial growth is that of sulfa drugs. Sulfa drugs are synthetic antibiotics which are called antimetabolites because they're going to prevent the metabolism of a particular chemical by bacterial cells. This is what happens. Bacteria can synthesize their own folic acid. Folic acid is needed to make nucleotide bases, which in turn are needed to make DNA and RNA. Without folic acid, a cell cannot replicate or can make proteins. One of the first steps in the metabolic pathway that synthesizes folic acid is a reaction that binds pyridine and PABA. Pyridine and PABA come together in an enzyme that will catalyze the reaction that brings these two reactants together. This is the first step in the metabolism of folic acid synthesis. PABA is one of the substrates for this enzyme that catalyzes the first reaction in the folic acid synthesis pathway. By the way, humans lack a folic acid synthesis pathway. Humans cannot make folic acid. Humans must consume or eat their folic acids in their food. So this metabolic reaction is exclusive to bacteria. Sulfa drugs have a conformation that is very similar to PABA. This is the chemical conformation of para-aminobenzoic acid, or PABA. This is the conformation of sulfonamide, a sulfa drug. Sulfa drugs are synthetic antibiotics. Look how closely they resemble each other. Sulfa drugs are competitive inhibitors for PABA. When a bacteria is given a sulfa drug, the sulfa drugs competes with PABA for the active site of the enzyme in the folic acid synthesis. Once the sulfa drug takes the place of PABA, the enzymes are inhibited and the cells cannot make folic acid. Notice that a competitive inhibitor must be similar enough to the substrate to fit in the active site in place of the substrate, thus blocking the site. Without sulfa drugs, PABA fits inside the enzyme and the cells can make folic acid. With sulfa drugs present, Now the sulfa drug can compete with the active site, with the substrate for the active site of the enzyme. If the sulfa drugs bind to the enzymes will be inhibited, and the cells, the bacterial cells, will not be able to make folic acid. Sulfa drugs are the first synthetic antibacterial drugs that were developed. Sulfa drugs were developed around 1932, a good 20 to 30 years before penicillin was available. Sulfa drugs are competitive inhibitors for the enzymes in the folic acid synthesis pathways. Sulfa drugs prevent folic acid synthesis, and bacterial cells die without folic acid. You should now be able to answer the following questions. Humans lack a folic acid pathway. We have to consume folic acid from our foods, like broccoli. Folic acid is needed for the synthesis of nucleotides, DNA, and RNA. Are sulfa drugs good antibacterial drugs? Do they have good selective toxicity? Can sulfa drugs harm human cells? Fluoride and other heavy metals like silver are non-competitive inhibitors, also called allosteric inhibitors. What is the purpose of putting fluoride in toothpaste and in drinking water? Why is non-fluoride toothpaste recommended for small children? Is fluoride a product with good selective toxicity?